You just gave my talk. Not at all. Thank you for a very kind introduction. So I'm going to tell you a tale today about two different potassium channels, the two that you just mentioned to you, IKR and IKS. I would tell you about my recent research, but I haven't done anything for 8 years. So I figured it doesn't matter how old. Am I okay here? Okay.
Yep.
So I'm going to talk about old stuff, whether it was my newest or my oldest. So I'm going to go back in time. And this is, to me, sort of a, uh-oh, it's not working. There we go. So to me, it's sort of an archaeological dig. I'm going to go back in time and dig out some of our early results and then try to bring you up to speed to current findings in the field of delayed rectifier potassium channel physiology, pharmacology, and biophysics. And that will obviously mean a lot of work that I haven't been involved with at all, but have been done by others. And the tale is divided into 3 chapters, if you will, 3 different topics. I'll first tell you about cardiac delayed rectifier K current as described by early voltage clamp studies, and about congenital long QT syndrome and how it's caused by loss of function in these 2 different potassium channel genes, and then about acquired or specifically drug-induced QT prolongation, sometimes called acquired long QT syndrome. It's really not a syndrome, it's just prolonged QT and arrhythmias which occur in response to that. So the first topic, this story starts really in 1962 with work done by Dennis Noble. Since he started at University College in London, then went to Oxford, and he was modeling Purkinje fiber action potentials based on Hodgkin-Huxley equations used to describe action potentials, electrical activity in the SWID axon. And that work was done in 1952. And Noble came along and extended this idea to cardiac tissue and could fully describe the shape and frequency of movement, if you will, of these action potentials with just 3 major conductances: the sodium conductance, GNA, you see here, and then potassium conductance here. And then in addition, there's a leak conductance. And just that alone, he could recapitulate these action potentials. And that was actually pretty amazing. It turned out to be completely wrong, but at the time, it looked really good. And in the potassium conductance, there's actually 2 components in this model: an inward rectifier current shown here, and a delayed rectifier current shown here. And rectifier just refers to the current being passed in one direction more easily than another. So inward and outward delayed rectifier in the outward direction. And for the electrophysiologists in the group, which are probably 3 of them left here, so this axis is reversed, but that's the way they used to plot them, believe it or not. And the delayed component right here is what I'll refer to as IK, or the delayed rectifier K current. And again, it's called this because if you look at the initial phase of activation of the current, there's actually a little delay as the channels go through multiple closed states before they open, and then in some cases inactivate. So that's the delayed part. And it's an outward rectifier because it preferentially passes current in the outward direction, not the inward direction. And then in terms of actual experiments, one of the very first studies was also done by Dennis Noble, but in collaboration with a postdoc with him at that time, Richard Chin, who went on to great fame mainly in neurophysiology. And they characterized this K current here, and this is the voltage clamp experiment where the steps in voltage are shown here. Just for one step I'm showing, and here's the corresponding current. You can see it's outward. You can't really see the delay here because this is a very long pulse. But upon repolarization to -30, you can see a different current, decay. That's called a tail current, or the deactivation of the current that's activated during the depolarizing pulse. And they did these experiments in sheep Purkinje fibers, which is a multicellular prep, which led to some problems. But what they described in this initial paper was both the onset of the current and the tail current, deactivation of the current, was composed of 2 components, 2 components defined by the rates at which these things develop. So there's a fast time constant and slow time constant for the activating current, and that's supposed to be IX1, sorry, a fast and a slow component for the tail currents. So two different components based simply on kinetics, and they describe these as potentially, or most likely, two different types of channels. And they called it IX instead of IK, like I just described to you earlier, because they didn't know if this was potassium selective channels or not. In fact, based on reversal potentials and other biophysical measures, it suggested that it passed at least the IX2 component, the slow component, passed sodium ions just as easily as potassium ions. And that was, and therefore just used the term X. I'm sure Musk was pretty upset about that later, but took his X away. So the slow component, IX2, is probably an artifact of measuring currents in multicellular preps. So Noble used these Purkinje fibers on multicellular prep, 2 microelectrode voltage clamp technique, but subsequent labs also used sucrose gap voltage clamp on trabeculi, which again are pretty large tissues. And this led to a variety of problems. Most importantly, that the membrane voltage is not adequately space clamped. In other words, you want to be able to change the membrane potential in all the cells all at once. In a multicellular prep, that's almost impossible. So you have all these kind of weird currents occurring within a tissue that you don't see in a single cell. And then in addition, you get potassium and or sodium ions that accumulate in restricted extracellular spaces between cells during depolarizing pulses. And that changes the reversal potential of the current, which makes it look like it's a non-selective channel, both sodium and potassium. This was followed up by studies in a number of different labs all over the world, and they all found only one delayed rectifier in ventricular muscle or Purkinje fiber, even using multicellular preps. In some cases, much smaller Purkinje fibers, but nonetheless, a single component, not two, not IX1 and IX2. And then in the early 80s, the voltage clamp technique was really advanced forward with whole cell patch clamp technique, where you could put a pipette onto a membrane of a single cell, blow out that piece of membrane and have access to the intracellular componentry, but also have a wonderful spatial clamp so you don't have that problem of multicellular preparations have. And also there's no ion accumulation or depletion. And you can control the intracellular contents by whatever you put in the pipette will now be inside the cell. And so this led to much more accurate and precise measurements of ionic currents. And for example, in rabbit sinoatrial node recordings, this current shown here was called IK by Shibasaki in Japan. And you can see the currents activate pretty rapidly, have a little bit of what looks like inactivation, in other words, a decrease in current during a pulse. And upon repolarization, the tail currents deactivate relatively rapidly. And they're half activated right around minus 22 millivolts. So this is a conductance voltage relationship. Just tells you how many, what percent, if you will, of channels from 0 to 100% are open as you change the membrane potential. So that was the current, the one current that was found in these cells. And then in another vertebrate, in this case, bullfrog atrium myocyte, the researchers, this is Joe Hume, who I knew very well back then, recorded a very slowly activating current, sort of like that IX2 that Oxford group had shown. And again, this is during a pulse duration of 5 seconds. So these currents are still activating during 5 seconds, where this is only a half a second and you're fully activated within 100 or 200 milliseconds. And these channel currents activated a more positive potential, minus 15 compared to minus 22. So again, a single K current in each cell type with widely different properties depending on the species and perhaps the tissue. So I think kind of the next big advancement in delayed rectifier K currents was trying to find agents that could specifically block them. Then you can start dissecting out the properties of the channel. And in the 1980s, in the late 80s, big pharma companies like Merck, which is where I was at the time, sought and then discovered a variety of new, more potent so-called class 3 antiarrhythmic agents. And this is just a quick refresher for those who know this sort of esoteric pharmacology. Antiarrhythmics are either class 1, 2, 3, or 4 based on their main mechanism of action. Class 3 prolong action potentials, and existing drugs like amiodarone and sotalol were typical of these, but they were not very specific. They had effects on multiple channel types and other receptors. So again, there was a search for new compounds, and this led to discovery of several compounds with IC50s in the range of 10 to 100 nanomolar, and were highly selective for IK. You take 10 to 1,000-fold higher concentrations before you affect a different channel type. And the three main ones at that time were E4031 from Eisai company in Japan, Defitelite from Pfizer, and MK499 at Merck. That was one of the compounds that my lab, obviously with the chemists who made the compounds, discovered in the late 80s. And all these compounds are methane sulfonanides, and that's the primary pharmacophore for these compounds. So methane sulfonanilide, that's what they have in common. And then the R group to the right there could be varied quite a bit, and would change the selectivity as well as the potency. So one of the first things I did when I was at Merck was characterize this E4031. We knew it was a K channel blocker just from other work, but I decided for whatever reason, I don't even know why, to look in guinea pig ventricular myocytes as the first species. And when you do that, you're looking at just the action potential, you see a prolongation of the action potential. This is probably about a micromolar concentration, so consistent with a class 3 effect. And if you then went to voltage clamp to measure the ionic currents that underlie these sort of changes in action potential, you see records just as shown here. So this is a very long pulse done at two different acquisition rates. So it's 10 seconds, and this is only 750 milliseconds here. During the 10-second pulse at plus 10, you can see that it's very slowly activating current, sort of like that IK in the bullfrog atria. And then put on this drug, and you see a small effect. The currents are blocked or inhibited both during the pulse and during the deactivation phase here. And this is the difference current here. And so the first idea was that, well, this must just represent voltage-dependent, incomplete block of a current. That's what we thought was going on. But further analysis by me and my wonderful technician at the time, Nancy Yurkowitz, decided, well, what if it's not that? What if you're actually dissecting out a different channel type? So there's two. There's one, the slow current, which isn't affected by the drug, and something faster, which is a different current. And that's what we decided was happening. And here are the difference currents of different test potentials, minus 30 through plus 40. You can see, especially at the high potentials, the currents activate extremely fast within a few milliseconds, but are smaller at the higher potential compared to these lower potentials. And if you plot the amplitude of the currents during the pulse as a function of test potential, you get this bell-shaped current voltage relationship here. And this is a little unusual for potassium channels, since most potassium channels, especially outward rectifiers, would just have currents that get bigger as the potential gets more positive. And this difference, I won't bore you with all the details, but is due to the channels going into an inactivated state after they've opened at higher potentials. So inactivated state is just a different type of closed channel configuration that doesn't allow conductance of ions. So you have closed, open, and inactivated. So that was sort of unusual and didn't really match anything that was published at the time. And the time constants for both the activation and deactivation phases of current, either opening or closing, are shown here. You can see that the fast time constant, there's also a slower component, which isn't shown here, varies from about 10 milliseconds up to about 170 milliseconds. So it's a pretty fast current. In contrast, the current that wasn't blocked by the drug, which I call IKS of time because it's slowly activating, is shown, just properties are shown here. And this is the shorter pulse at potentials from minus 20 to plus 40 before drug and then after drug. And the non-initiated looks like it's the same.
Oops.
Okay.
This one.
I don't know what you're doing here.
This one. You can use this one.
Oh, I
Yeah,
had
do
it
the
used.
plan.
I'm an old man, I do this. So that, but anyway, they're actually different. As I just showed you, if you look at a difference current, the currents are quite different. But this would be called IKS, and activation time constants are much slower, 1 to 2 seconds instead of 10 to 170 milliseconds. The deactivation or channel closing is similar to that other current. So we have two currents, IKR, the rapidly activating, and IKS, the slowly activating current. And then if you give long enough pulses to characterize both channels at once, you get the activation curve shown here as IK total with filled squares. And then add the drug, take this subtraction difference, and then to define the voltage dependence of the fast component and the slow component, you can see there's quite a bit of difference, about 38 millivolt difference in the half activation voltage. So and again, because these properties looked like a single component in either case, I concluded that these are two different channels. One's drug sensitive, the other's not. And this just compares the current voltage relationship for IKR and IKS. IKR I already showed you, it's kind of bell-shaped. This is mainly due to inactivation. IKS just keeps getting bigger and bigger, like a typical delay rectifier described in the literature. And the total current is like this. And you can see just from that, a lot of people who are just studying the current without any drugs to dissect, you would just say, oh, there's a little hump there, but it's basically a single current going outward. And I should say that the interpretation that there are two different channels was initially not well received. I should say a lot of people said, no, you're just looking at voltage and block. That's not two different channels, which is a fair criticism. But we interpret it differently and said IKS predominates with long pulses and at higher voltage. You can see IKS is big here, IKR is small because it inactivates. So that made it pretty clear to us that the guinea pig fast, rapid delayed rectifiers is basically equivalent to what was measured earlier in cytometry load from rabbit. And you can see that both in the IV relationship, they're both bell-shaped. V1/2 activation was similar, and time constant of activation, 0 millivolts, was basically the same, as was the single channel conductance. The slight difference here is because this was recorded in higher extracellular potassium than we used. But at any rate, IKR was basically the rabbit delayed rectifier. And this really slow current you see in frog, we looked very much like IKS for guinea pig. So I would conclude from that, the Noble and Chen were correct, even though all these papers for about 15 years afterwards said they're wrong. When we came along and said, no, I think he's right, and this is why. But it wasn't proof, again, because we didn't have, none of these channels were cloned or anything, and we just had these electrophysiological experiments to rely on. So now I'm going to switch gears and talk about chapter 2, congenital long QT syndrome, and limit my discussion just to the loss of function mutations that occur in the genes that encode these two different channels. But first, a little bit of overview of what long QT syndrome is. It was first described in 1957 by Javel and Lang-Nielsen in a very large Norwegian family, and was characterized by not just a long QT, but deafness, recurrent syncope. They were fainting a lot of times, and that was during an arrhythmia, and quite often sudden death in a number of the family members. And it was inherited in an autosomal recessive manner. And then later in time, like 5 or 6 years, 7 years later, Romano and Ward separately described patients with normal hearing, very long QTs, recurrent ventricular tachyarrhythmias, but these were inherited in an autosomal dominant manner. The estimated incidence of long QT, regardless of type, is about 1 in 2,000 in the general population. About half the affected, or the gene carriers, show no symptoms whatsoever. So it's pretty good penetrance, but not complete. And again, for those who aren't familiar with electrophysiology. The QT interval is just taken from the body surface ECG, where you just measure between the Q wave, this wave here, and the end of the T wave. And that basically represents repolarization in the ventricle and the ventricular cells. And a long QT, where the T wave is prolonged, is defined as a heart rate corrected QT interval greater than 440 milliseconds for men or 460 milliseconds for women. So they have a longer baseline. And again, as I just mentioned, the T wave really just represents prolongation of the ventricular action potential throughout the tissue. So when an action potential is prolonged sufficiently, sometimes you can generate in a single cell these so-called early after depolarization. So like little mini action potentials that occur before final repolarization has been allowed to occur. And Joram Rudy and others describe these EADs as being due to a reactivation of the L-type calcium current as shown here. These calcium currents are inward currents that are going to prolong the action potential and allow for the upstroke of these little mini action potentials here. And EADs can trigger the onset of an arrhythmia called torsade de pointe, especially in the setting of abnormal spatial dispersion. So just differences in how long repolarization is in different parts of the heart, like Purkinje epicardium versus mid-myocardium and endocardium. And especially if in abnormal dispersion of ventricular repolarization. So these regions of the heart can, under normal conditions, have different repolarization rates, but under pathological conditions, some are going to repolarize faster than normal, some are going to be longer than others. So that's called a spatial dispersion of repolarization. And that sets you up for these arrhythmias. And the arrhythmia looks like this. So this is an ECG tracing, a continuous in a single patient, heart rate shown here, the normal ECG right there, QRST. And then quite often a torsade is initiated after a pause, a very brief pause in the normal rhythm. You can see the arrhythmia starting here, back to normal sinus rhythm for a few beats, and then back into the arrhythmia. And then it sometimes can go into this sort of rhythm. This is the torsade de pointe, which means twisting of the points, or it's really the sinusoidal twisting of the QRS axis around the isoelectric line of the ECG. So it's a very characteristic type of arrhythmia, ventricular arrhythmia, associated with long QT syndrome. This can defibrillate, can degenerate into ventricular fibrillation, and that's what leads to sudden cardiac death. Just as often, though, the torsade can revert back to normal sinus rhythm, and the patient just feels has a fainting incidence. So, unlike what Dennis Noble first modeled, we now know that the ionic currents that underlie ventricular human ventricular action potentials is includes many more currents than initially thought. There's an inward and sodium current and an inward calcium current, which are responsible for the initial upstroke and the start of the plateau. Again, inward currents, these are going to raise the membrane potential. All of these outward currents here are going to cause repolarization of the action potential. There's a lot more currents in that, and are ion channel currents in that, and including these three different ones: ultra rapid delayed rectifier, which I'm not going to talk about today. It's KV 1.5 channels, acetylcholine activated K channels, funny current, and P-type calcium currents, which are found in atria but not ventricle. And then under ischemic conditions, you can have an intracellular sodium activated K channel or an ATP depleted K channel activated. So there's lots of different channel types. And back in the late 80s, early 90s, the most common assumption for the mechanism of long QT syndrome was that it must be caused by mutation in ion channel genes. It didn't have to. It could have been a lot of different genes that were in some way responsible for repolarization. Specifically, anything that would increase inward currents like sodium or calcium, for example, a gain of function in these channel function or a loss of function of potassium outward currents would lead to a prolongation of the action potential. So any of those things are possible. And it was about this time that I moved from Merck to University of Utah in 1993 or two, I can't remember exactly. That I met Mark Keating, who was in cardiology and a molecular geneticist working on long QT syndrome, and they were trying to map the different gene locations for this disease. And we go on bike rides or hiking or cycling almost every day. And we talk about, mechanism, what could it be? And he said, well, any fool who knows about this stuff will tell you the loss of function of one of these currents or gain of function of one of these will cause long QT syndrome. The genes that encoded these channels were known. These were not, I should say. But still, there was no proof. That's what it was. It was just kind of the obvious is what would do it. And then an amazing period for the University of Utah was in the mid-1990s when the first genes for long QT syndrome, in fact, any inherited arrhythmia in humans, were discovered in Utah by Mark Keating, who led the lab in collaboration with Mike Vincent, a cardiologist, and an amazing clinical coordinator, Catherine Timothy. During this period, long QT syndrome loci were mapped to 3 different chromosomes, chromosome 11, 7, and 3, and based on the chronology, they were there for the name of LQT 1, 2, and 3. And then in very rapid succession, just in 2 years, using a positional cloning approach, Mark and his and study that was headed by King Wong, postdoc in the lab at the time, found that KVLQT1, a novel gene never been described before, was obviously a potassium channel gene based on sequence, but function was unknown. That was found to cause long QT type 1. And then using a candidate gene approach, just looking for, based on refined loci, which genes are present in that location, they discovered that hERG gene mutations, which had just been cloned, like I think that year, causes long QT and SCN5A, the sodium channel gene causes long QT3. And as I just mentioned, the SCN5A encodes the cardiac sodium channel, so it's a gain of function, just more inward current in this channel that led to long QT syndrome. Specifically, the channels didn't inactivate during depolarized potentials as well as they did under normal conditions. These 2 genes encoded novel K channels at the time. Clear they were K channels based simply on sequence, but the specific function was unknown. These are now called KCNQ1 and KCNH2, just because of a more commonly used naming protocol. But anyway, I'll try to just use one. And then subsequently, over the 20 years or so, 30 years afterwards, people started cloning all sorts of new genes associated with long QT syndrome. And if you look in the literature now, there's 17 different types of long QT. Maybe, I really don't agree with that. LQT 1, 2, and 3, the ones I just told you about, are for Romano-Ward syndrome, that autosomal dominant form. And autosomal recessive is caused by dual mutations in KCNQ1. And then the study that was pioneered by Louis Potacek as the geneticist and our friend right over here, and Marty found that KCNJ2 loss of function mutations causes Anderson-Tawil syndrome. This is a multi-organ syndrome that includes periodic paralysis, so it affects the skeletal muscles, and it is really a large U wave rather than a prolonged QT. It's actually just the U wave becomes more prominent, and it's the equivalent of looking at a long QT. And also Timothy syndrome, that's named after Catherine Timothy, gain of function mutations in the L-type calcium channel. And this is a really severe disease. It probably is probably lethal in utero quite often, but it's very severe pathology, includes autism, developmental delays, syndactyly, all sorts of problems. But since all these things were published, it's now clear that 4 types of these 8 long QT syndromes that are due to mutations in calmodulin genes are not really long QT. They're very severe arrhythmias, but it's an early onset arrhythmia that's not really like a typical Romano ward. So the thinking now is not to include these as long QTs. And these 7 different genes and LQT types are now disputed based on absence of good genetic or physiological data. So you're left now with really just these 3 and these 3 and maybe that one. So there's not that many genes that cause long QT syndrome. No, it's not that these are all probably associated with arrhythmia, but it's not long QT. Oh, and then in terms of which are the most important, 70 to 90% of long QT mutations are in one of these 3 with that breakdown. So these are by far, in a way, the most common causes of long QT. So as I mentioned, both of these key channels had unknown function at the physiological roles at the time of the long QT genetic findings. So my lab quickly set up, very quickly, a heterologous expression system where we took out oocytes from a Xenopus laevis African clawed frog, injected the single oocytes with RNA encoding the channel of interest, and used 2 electrode voltage clamp technique to record currents. And I think I've already mentioned already. So you apply steps changes in voltage and measure the resulting currents. So this is kind of an IKS current. So HERG, which has a crazy name, it's human EAG related gene 1. Now it's called a KCNH2, a much nicer name, was named because of a mutation found in Drosophila called ether agogo. So when you add ether to a jar holding some Drosophila flies who have a certain kind of mutation, turns out mutation in EAG gene, they go crazy, look like a dancer, so they call it ether agogo. And then Jeff Wonke and Barry Ganetsky in Wisconsin '94 homology clone based on EAG Drosophila using a human hippocampal cDNA library and pulled out something very related, this human EAG related. And most people still use this term. I'm going to use this mostly for this talk. It's really KCNH2 is the more official name for the gene, at least now. So it was a really exciting time because I had studied IKR and IKS in myocytes, and here we literally on 5 minutes into the first experiment, in oocytes that were expressing KCNH2 or HERG, we got these currents here. And I immediately knew within minutes, I go, gosh, that's IKR. That looks exactly like IKR. And in the usual way of describing it is that the currents are slow to activate at these negative potentials. And then you get to about 0 millivolts, the currents reach a maximum. And then 10, 20, 30, 40, 50 millivolts, the currents get smaller, the tail currents get bigger, just like IKR did. And if you plot the amplitude of the current at the end of the pulse, you see current voltage relationship, and it's bell-shaped, just like IKR in myocytes, or the IK and the synovial node from rabbit. And if you plot the amplitude of the tail currents as a function of these test potentials, you get the activation curve I've been talking about. And again, with a V1/2 that matched IKR in myocytes. And I won't tell you how, but it's relatively complicated. But if you do a data transformation on the amount of decrease in current you get as a function of voltage, you get the voltage dependence of inactivation. So this tells you that at minus 120, no channels are inactivated. At plus 40, they're almost all inactivated. In other words, no current is passed. And it's this inactivation relationship that results in an IV that has a bell shape. And this is really important because HERG or IKR inactivates at plateau potentials is what allows a ventricular action to have these long and delayed phase of repolarization. If the current's activated from closed to open and just stayed open, this is supposed to be potassium ion going through the channel, plateau phase would be very short, and you get an action potential, it repolarizes it right away. But instead, the channels are primarily in an inactivated state, not in an open state at these high voltages, because they go through this little tiny transformation you can barely see here, where the selectivity filter collapses a little bit, doesn't allow potassium ions to go out, so it doesn't conduct current. So it's basically like a closed state, even though the configuration of the protein is very different than the closed state. And then upon repolarization, the currents recover from inactivation. Inactivation gets less as they go back to an open state. In other words, going from here to there. So this property is really important and defines the mechanistic basis of why you can have these long plateau phases in ventricular cells, especially. It's also true in atrial cells, but in atrial cells, the current action is a little bit shorter. We then went on to show that these methane sulfonanides, like the MK499, inhibits her current, just like an IKR. In myocytes, shown here, current at 0 millivolts after drug, and the dose response here at different test potentials. And the IC50 is about 120 nanomolar. That's in oocytes. In mammalian cells, it's more like 10 nanomolar, but oocytes have yolk inside, and drug tends to accumulate in there in this lipid phase. And so you always have a higher IC50 in oocytes compared to myocytes. So based on physiological, biophysical, and pharmacological properties, we therefore decided that hERG equals the cardiac IKR channel. Now, hERG, like all voltage-gated KV channels, are formed by co-assembly of 4 subunits. Each subunit has 6 transmembrane domains, shown here. The first 4 form the voltage sensing domain. S5 and S6, together with a 4-helix and selectivity filter right here, form the actual outline of the inner pore in which potassium ions and water stream through the channel and go out or in through the final pore. And this just shows a top view of a cryo-EM structure for hERG. Here's the pore domain. The voltage domains sit out like little wings out here. And this is the S6, this right here. S6 lines the inner cavity, and that's the selectivity filter, the little thing here. And that's the carbonyl oxygens from the amino acids in that position is what allows potassium ions, dehydrated potassium ions, to bind in different positions within the selectivity filter and then go out. And that's why these kind of channels are potassium selective. Just the hydrated or dehydrated radius of sodium is a little bit different than potassium, so it doesn't go through as well. hERG is actually, at least in real cells, composed of 2 different subunits. hERG1A, which is the full subunit, including this very, very long N-terminus, and hERG1B, which is alternatively spliced variant, which has a very short N-terminus. And done due to really wonderful work by Matt Trudeau and Gail Robertson over a number of years, who really pushed the idea that 1B is really important. It's very clear now that IKR channel is actually hERG1A and 1B heteromers, and how much of 1A and 1B I think varies a lot, but it's clear that you need both of them to have the exact properties of IKR that you see in a myocyte. So there's many, many variants in the KCN2 gene now that have been described. These are all on the ClinVar NCBI public database. Not all of them are pathogenic, but these are just, and whenever a variant is found, that you put it in the database, and some almost 600 are pathogenic. Not all are long QT, but most of them probably are. 218 are likely pathogenic, and then a whole bunch of uncertain buses, they're called, uncertain significance, and then a bunch that are benign or likely benign. So there's been a huge effort to characterize in vitro technology used to find out if these likely pathogenic or uncertain significance changes are actually pathogenic. So this just shows a kind of a partial example of where all the different mutations can be in a single subunit. And the only point of showing this is that they're all over the place. They're not concentrated in one area. Anything obviously that's in the pore is probably going to be more detrimental or in the voltage sensor, and that's where you'll see a lot of different mutations, but they're also in the N terminus and the C terminus. There's a lot of mutations. And so I just give you one example of the physiological dysfunction of some of these mutations that we studied quite a long time ago, but in this case by Fuda Wang and Jun Chen. And this is 3 different mutations, all very close to each other in the pore helix, which is this feature right here. And they're just, again, point mutations, single amino acid changes. So here's a wild type current in oocytes that were injected with 10 nanograms of cRNA, and quite a bit of RNA. You get really big currents. And if you half the amount of RNA, you get about half the current. And that's just a wild type alone. If you look at the mutant subunits alone, you get 0 current for 612 or 613 mutations. Again, this is with 30 nanograms, so 10 times the amount of current, amount of RNA used to generate these currents. So really huge loss of function. I mean, 615 gave little tiny currents, but they were still there. But that in patients, there are no homozygous mutations like this. They're all heterozygous. I'll show you in a moment. So anyway, the loss of function you see with expression of the mutant subunits alone is due almost entirely to defective trafficking, shown here by HA tagged constructs, where you just measure how much of the protein gets to the cell surface. Here's wild type uninjected, and you can see with these mutant subunits, some proportion, but a very small proportion of the channel subunits or channels get to the cell surface, and that alone can explain lack of current resulting from expression of those mutant proteins. And it's now clear that, at least for her, about 90% of the missense mutations are due to a disruption in channel assembly or trafficking. This is mostly work done by Craig January's group, also in Wisconsin, and they published a lot of papers, and that's been very revealing. And so a lot of the defects you can see in gating of mutants might be interesting to note, but in fact, most of these mutations just screw up assembly and or trafficking to the cell surface. So for these 3 mutants, the next thing we do is to co-express the wild type and mutant subunits together to mimic this heterozygous condition in patients. So here's the wild type currents, and here's 5 nanograms of each, so twice as much as twice the amount of RNA used for the wild type. You see the currents are much smaller. We did that for all of them, and you can see here that if the currents were the same size as wild type, that would be a haploinsufficiency condition where the mutant channel just doesn't do anything on its own, just disrupts, disrupted folding goes away. But if a disrupted protein can co-assemble with the wild type, then you're going to get some dominant negative effect. And that's what you're seeing here, for at least for these 2 mutants. But it's a rather subtle effect. It's not a huge, it's mainly probably a trafficking defect with some dominant negative interactions as well. And so, and again, hundreds of these mutations have been looked at now, and in all cases, LQT2 is due to reduced rapid delayed rectified K current. Just one component is reduced that prolongs the action potential, and that can lead potentially to early after depolarization, which was triggered with Torsades de Pointes, that arrhythmia that's so well associated with this disorder. And again, arrhythmia is most likely to occur in setting of an altered substrate like hypokalemia, just low serum potassium, or if the patient is taking a drug that blocks hERG, which as I'll show you later, is very, very common. So KVLQT1 was also a novel K channel. An expression study revealed right away that the currents were not recognizable to us. There was some, had unusual properties. It didn't match anything in cardiac tissue, including this little funny hook here, which is not super important, but it shows that there's some inactivation occurring in these channels. And the V1/2 was, again, didn't match anything we'd seen in myocytes. And it wasn't totally potassium selective. There was some sodium conductance as well in these channels. We had already known at this time that there was this current or this channel subunit called min-K, which was short for minimal potassium, which is a 130 amino acid single transmembrane domain protein that, when expressed in xenophagocytes, but not mammalian cells, shown here, produces a current that looks just like IKS. So it doesn't look like a K channel. I mean, now it seems kind of silly, like, well, that couldn't possibly be a channel. But at the time, again, right before all these discoveries, one group, prominent group, suggested that these subunits actually just co-assemble in a large aggregate, and that somehow gives you these currents that give you this extremely unusual property, very slowly activate. No other K current looks anything like that. The alternative interpretation, of course, was that min-K is just a beta subunit that combines with some unidentified KV subunit that everybody was thinking about. And when KCNQ1 was cloned as a novel K channel gene, we go, that's the obvious candidate. And again, I think within days of that, we did that experiment in the mammalian cells and showed that min-K alone doesn't do anything, just like what other people had reported. KCNQ1 gives you these little currents. You put the two together and you get these huge currents. They're very slowly activating. And this association increases current mainly by increasing single channel conductance. Just the amount of ion flow in a channel is much, much greater in the heteromer than in the homeric KCNQ1. And obviously slows the activation rate, increases PIP2 sensitivity, removes the inactivation. You no longer see the little hook in the tail. And the currents were inhibited by a known IKS blocker, chromanol 293B, and some benzodiazepines, which were cholecystokinin receptor antagonists that we had discovered at Merck years earlier that either activated or blocked the channel. They did the same thing to this KCNQ1 and min-K. So again, based on biophysical properties and pharmacological properties, it was decided that these heteromultimers are responsible for the cardiac IKS channel. And I should note, I forgot to mention on the other one, with KCNQ1 and min-K, these experiments were done almost identically by a French group at the exact same time as us, and we both published back-to-back papers. We didn't know the other one was working on it. We submitted our paper, was accepted, and said we can't publish it for 3 weeks. And I'm like, okay, whatever. And then we found out why. Jacques Brahanen, was a friend of mine now, had published the exact same thing. So everybody had the same idea. Nothing is new in science, right? It's all derivative. So the cryo-EM structure has now been published by Rod McKinnon's group out of Rockefeller. And that shows not only the configuration of the proteins, the 4 subunits, the core domain and the voltage sensor domain, but also the location of these min-Ks or KCNEs. He actually did the structure with KCNA3, but it's the same as KCNE1. So these binding of these little tiny proteins here really change the biophysical properties of the channel in very dramatic ways. And they combine with 1 to 4 min-K subunits. There's a huge argument whether it's 2 or 4. I won't get into that. And there's evidence for both interpretations. So the Q1 gene, there's again thousands of variants reported. Many of them are uncertain significance. KCNE1 is much smaller. Much less mutations have been reported. 70 are considered pathogenic. Only a few of those are LonQ-T5 associated disease mutations. So they're just others are just considered pathogenic based on how much dysfunction the mutation causes in the protein. LQT5, due to E1 mutations, has very low disease penetrance and mild phenotype compared to Romano Ward LQT1. And I'll just give a quick example here of 2 different mutations in the C terminus of min-K. Again, this is work done by Marty, bringing back old memories, isn't it? And Igor Splowski, who is the geneticist involved. Marty did all the physiology. And so here's the wild type channel, co-expressing wild type KCNQ1 plus min-K. And this is the mutant min-K. And you can see the currents are much smaller. So the current amplitudes are very much reduced by this mutation. In addition, the voltage dependence of activation is shifted to more positive potentials, and the deactivation rate is faster. And these two effects will also decrease current magnitude in addition to just the effects on total current. So there's three different mechanisms there. And the exact same thing is going on with the other mutation, the serine to leucine at position 74. It's just less prominent decrease in current, but the shift in activation, the increase in deactivation rate are all the same. And again, as I showed you with IKR, LQT1 or 5 long QT syndrome is due to reduced IKS, action potential prolonged, EADs probably subsequently occur, and that triggers Desaad-Dupont. I've only got a few minutes left, so I won't be able to go through this, but I'll just quickly go through it. Acquired drug-induced QT prolongation due to block of hERG channels. We spent a lot of time on this in my lab, and then also characterizing agonists, which might be a good therapy for a long QT syndrome. I'm not going to tell you about any of that stuff. That's probably 10 years of work, but I'm going to talk about it. But drug-induced QT prolongation is most often caused by an unintended block of hERG, and almost never, I wouldn't say never, but very, very rarely due to other potassium channels or other channels at all. It's caused by a huge number of structurally diverse compounds from multiple therapeutic classes, and it's a major safety issue in drug development, as you might guess. In this drug-induced QT prolongation led to the withdrawal of 12 different drugs, some of them shown here, and then a whole bunch of other drugs are identified as known risk, possible risk, or conditional risk. And that represents known risk is 4% of all drugs on the market, and possible or conditional risk represents 15% of all drugs on the market. So it's a huge problem that's dealt with both clinically and also during drug development. And then there was this organization of mainly drug company people, but also physicians, that decided to put out these guidelines that, including the requirement, or highly suggested, I should say, all compounds should be subjected to hERG safety screening, and 60% of drugs in development show this liability. And it's been about a $2 billion business a couple of years ago. Well, we wanted to know, why are hERG channels so susceptible to drug block and not other channels? So the first thing we did is show that the currents have a kind of a unique response to the drug. And the key experiments shown here: measured current under control conditions, done by Peter Spector, who was a cardiology fellow at the time, and then add the drug for 10 minutes, don't pulse, and pulse again, and you can see there's virtually no effect. In fact, the currents got a little bigger. But if you then repetitively pulse, just so-called use the channels, then you see a much bigger block. You could also get block using this same protocol, but hold at minus 50 millivolts instead of minus 80, so a more depolarized potential, and you get a similar block. And again, if you look at the activation curve, minus 80, all the channels are closed. At minus 50, there's a slight number of channels that are open. So the interpretation here would be that channels that are blocked by the drug require that they be opened first. And the key experiment for that is shown here, where we measure currents at a really long pulse, almost 20 seconds, put on the drug, wait 10 minutes, a huge concentration of drug, 10 micromolar, and you get that. So the initial current's identical, but now the channels are opening, the drug gets access to the central cavity and blocks the current. So it's an open channel blocker, requires the channel to open before the drug can block, and suggests that the binding site is located in the central cavity. And because of time, I won't go through all this. I'll just go through the main findings. We did alanine scanning mutagenesis of all the residues in hERG that were known at that time to potentially face the central cavity, and did one mutation at a time and identified a bunch of residues that look like drug binding sites, because these mutant channels were hardly blocked by the drug, even though it's just a single mutation. done by John Mitchison. We did this with other drugs. It's a very similar pattern. This shows a docking of the drug inside the central cavity and these key residues shown here. These two residues are present in all channels, so that we thought they're probably not really important, but these are pretty unique. I won't show you that. And this is the sequence showing that the tyrosine and the phenylalanine, which are key drug binding sites, are different residues in all the other KB channels that are expressed in the heart. And I'll just skip this and then just go to the final bit of data that's now published. Now that we have the cryo-EM structure of HERG, none of this is available when we were doing the work. We just had bacterial channels at the time. And it turns out that HERG has this very unusual feature, right? Below the four helices and between these four helices and the S6 transmembrane domains that line this central cavity, there's little hydrophobic pockets. It's going to be 4 of them because these are homotetramers. And it turns out the drugs, first of all, the drugs bind in here. And most importantly, these side pockets are not present in any of the other KV channels in the open state or when HERG is in a flow state. So it's evidently these things are really key. In the time this cryo-EM structure was published, they couldn't get a drug in there and get an image. But since then, other groups have, out of Furutani's group in Japan, and you can see it fits in that little space. And this is, again, showing E431 in these 2 pockets, shown from above, right in here. So it's these little hydrophobic pockets that allow these drugs to bind. Here's another drug doing the same thing. So just in summary, there's 2 types of delayed rectifier K currents. IKS activates slower than IKR. With rapid stimulation or adrenergic stimulation, it gets bigger. And I like to think of it as kind of a tortoise and hare. You know, this IKR activates really fast and gives you fast repolarization. But if you give long enough pulses or multiple pulses, the old tortoise catches up. Sort of is a perfect example of what Dan Roden called repolarization reserve. Molecular mechanisms of long QT, functional characterization mutations obviously enables risk stratification and rationale for new drugs. And again, this is a huge effort we put in. I'm not talking about today, but we found all kinds of activators. And in terms of the pharmacology, a promiscuous drug binding is very much like multi-drug receptors or transporters, in that there's key residues which are polar, which can interact with polar regions of a drug, and then these highly hydrophobic residues which allow pi-pi stacking or cation-pi interactions, as well as these substrate pockets which are not found in other K channels. I mentioned all the names of the people who are in blue. This is the whole group over the years. And I'll just end with showing the motley crew over the years. So thank you so much.